Hi everyone, my name is Katie and I'm a fourth year student in biological sciences. This past summer, I had the opportunity to work on a project in Dr. Jocelyn Bosson's lab in the Faculty of Veterinary Medicine, where I aim to study the association between strongyle parasite infections and overwinter survival in the Sable Island feral horses. Wild animals are typically infected by numerous gastrointestinal parasites that significantly impact their health and fitness. Among these parasites, some are more pathogenic and thus pose a greater threat to host survival. For example, feral or wild horses can be infected by over 40 different species of a parasite known as strongyles, and there are two main groups that these species can be classified under, and that is small non-migratory strongyles, which live within the host's gastrointestinal tract, and large migratory strongyles, which migrate extensively through host tissue such as blood vessels and cause severe internal damage, meaning they are particularly harmful. However, identifying gastrointestinal parasite species non-invasively in wildlife species using traditional corporological techniques is challenging due to morphological similarities between the eggs of different species. In this study, I applied a molecular technique known as DNA metabarcoding to identify and characterize mixed species parasite infections in the feral horse population of Sable Island, Nova Scotia. My goal was to investigate the relationship between large migratory and small non-migratory strongyles and the horse's probability of surviving the winter. With my study, I hope to improve the understanding of host parasite dynamics in wild populations and demonstrate the value of utilizing molecular techniques in these systems. Sable Island National Park Reserve is a 49-kilometer-long sandbar located about 300 kilometers off the coast of Halifax, Nova Scotia. It is home to a population of feral horses that were first introduced in the mid-1700s and have since been the only large vertebrate mammals on the island, with population numbers fluctuating between 150 and 550 individuals. These horses have been the subject of a long-term ecological and evolutionary study since 2007, which includes yearly population censuses and sample collections during the field season, which is from July to September. For my project, I analyzed parasite infection data obtained from 2,822 samples collected from 946 individual horses over 9 years. To obtain this data, fresh fecal samples were first collected in the field alongside individual data including age, sex, and location. The fecal samples were then examined under a microscope to count the number of strongyle eggs. These eggs were incubated for 6 days to reach the larval stage, after which DNA was extracted from the larvae. Using DNA metabarcoding, we were able to identify the strongyle species by detecting species-specific DNA sequences in each larvae. The parasite infection data was then categorized into large migratory and small non-migratory strongyles, and generalized linear models were used to assess the association between these infections and overwinter survival probability. This model also accounted for factors such as age, sex, year, and location. The results of my analysis showed that in Sable Island feral horse population, small non-migratory strongyles do not have a significant positive or negative association with overwinter survival. On the other hand, large migratory strongyles have a strong negative association with overwinter survival. This means that as the level of large migratory strongyle infection increases, the probability of horses surviving the winter decreases. These findings are intriguing not only for the Sable Island feral horse population, but also for wildlife health studies more broadly. While my research demonstrates an association between large migratory strongyle infections and decreased overwinter survival probability, future studies could explore the underlying mechanisms behind this association. Overall, this study highlights how advancements in molecular techniques can enhance our understanding of host parasite dynamics in wildlife populations, which is vital for wildlife conservation efforts. Thank you for watching this presentation. I hope you enjoyed it and found it interesting. I also want to thank all members of the Poissant Lab for their continuous help and support, as well as the organizations that make the Sable Island Long-Term Study Project possible.